Hello Matrix. Do you ever wonder if there are really as many neurons in the human brain as stars in the Milky Way? This is a question that has fascinated both scientists and laypeople alike. It's an attempt to find a common point of comparison for two seemingly incomprehensible entities, the enormity of our galaxy and the complexity of our own brains. When we talk about large numbers, it's challenging to really grasp their magnitude. The human mind is not naturally equipped to comprehend things on such a grand scale. To give you some perspective, let's talk about the concept of a billion. A billion is 1,000 million. If you had $1 billion and spent $1,000 every day, it would take you over 2,740 years to spend all your money. Now, imagine you have not one, but hundreds of billions of dollars. That's an astronomical amount of money, isn't it? In the same vein, it's hard to visualize what 100 billion of something looks like. That's why science communicators often resort to analogies, like comparing distances to the length of football fields, mass to the weight of fully loaded 747 airplanes, or energy to the power of nuclear bombs. These references are something we can more easily understand and relate to. The comparison between neurons and stars is particularly fascinating because it involves two essential aspects of our existence. Neurons are the fundamental units of our brains, the physical structures responsible for our thoughts, emotions, and behaviors. Stars, on the other hand, are the building blocks of galaxies, including our Milky Way, and have been a source of human awe and wonder for millennia. By equating these two, we feel a sense of connection between the inner workings of our minds and the vastness of the cosmos. Now, let's delve into the specifics. For a long time, it was widely accepted that there were around 100 billion neurons in the human brain. But it's essential to clarify that this number was not definitively proven in a formal scientific setting. Instead, it was a broad estimate based on other measurements and interpolations. A landmark study in 2009 by Azedo and colleagues challenged this assumption. They used a novel method to estimate the total number of neurons in the human brain and arrived at a figure of approximately 86 billion neurons. While this is still an astoundingly high number, it's somewhat less than the earlier estimate. To understand how scientists arrived at this number, we need to delve into the world of neurobiology. A common way to estimate the number of neurons in the brain involves counting the neurons in a certain part of the brain and extrapolating this to the whole brain's volume. This method might remind you of how astronomers estimate the number of stars in the Milky Way by counting stars in a particular region and then extrapolating based on the galaxy's overall size. However, this method has its shortcomings. One issue is that the density of neurons isn't uniform across the brain. For instance, the cerebellum, a part of the brain located at the bottom and back, which is usually illustrated as purple in diagrams, contains about half of all neurons in the central nervous system. However, its volume is significantly less than half the brain's total volume. This discrepancy shows that some parts of the brain are packed with more neurons than others, making the extrapolation method somewhat inaccurate. Another issue is the sheer difficulty of counting neurons. Neurons are incredibly small and densely packed, making them tough to distinguish and count separately. The situation is further complicated by the fact that neurons are mostly transparent. Scientists often use staining techniques to make neurons visible under a microscope. One such method is the Golgi stain, named after the Nobel Prize-winning scientist Camillo Golgi. The Golgi stain only colors a small fraction of neurons, for reasons we don't fully understand. So when you're looking at a Golgi-stained slice of brain tissue, you're only seeing a small subset of the neurons present. The rest are invisible, making it challenging to count them accurately. In the image behind me, you might see one beautifully stained neuron with its complex tree-like branches, called dendrites, and long tail-like axon. But keep in mind that the visible neuron is just one among potentially hundreds that occupy the same space but didn't take up the stain and hence remain unseen. So, with the Golgi stain method, scientists estimate the percentage of neurons that the stain colors, count the number of stained neurons in a piece of brain tissue, and extrapolate from that. But this process introduces two sources of potential error: the estimated proportion of stained neurons and the counted number of neurons in the sample. Therefore, this method is not very precise. The researchers behind the 86 billion neuron estimate used a different method. Instead of trying to count neurons in a piece of brain tissue, they dissolved the cell membranes of cells within the brain, creating a kind of brain soup. They then used different markers to stain the nuclei of these cells, which allowed them to distinguish neurons from other brain cells, like glia. This method has a significant advantage. It doesn't rely on extrapolation from one part of the brain to the whole brain, which can be problematic given the different neuron densities across brain regions. Instead, by staining and counting the nuclei of neurons in the entire brain, they could directly estimate the total number of neurons. However, even this advanced method has its limitations. It still relies on extrapolation, albeit in a different way. Instead of extrapolating from a part of the brain to the whole brain, it extrapolates from the number of stained nuclei to the total number of neurons. Currently, scientists are exploring new approaches to more accurately count neurons. One such initiative is the Human Connectome Project's iWire game, a citizen science project that invites people worldwide to help map the brain's neural networks. It's hoped that such crowd-sourced efforts might eventually provide a more accurate number, reducing the reliance on estimation. So, are there as many neurons in the human brain as stars in the Milky Way? The current estimates suggest not. The latest figures for the number of stars in the Milky Way range from 200 to 400 billion, far exceeding the 86 billion neurons in the human brain. But who knows? As our scientific methods continue to improve, we might yet discover that our brains are even more densely packed with neurons than we currently believe. To wrap up, comparing the number of neurons in the human brain to the number of stars in the Milky Way is a powerful analogy, highlighting the astonishing complexity of our brains and the vastness of the cosmos. While the numbers might not match exactly, the sense of awe and wonder they inspire is a testament to the beauty and mystery of both the human mind and the universe we inhabit. The implications of these numbers are not just about the brain's complexity, but also about our place in the universe. When we talk about billions of neurons and stars, it's not just to show off big numbers. It's to give you a sense of scale and importance. The human brain, the organ that allows us to think, dream, and perceive the world, is as complex and intricately structured as our vast galaxy. This fact underscores the importance of scientific research and discovery. Understanding the brain and how its billions of neurons work together to create consciousness and intelligence is one of the great challenges of neuroscience. Similarly, understanding the galaxy and its billions of stars is a monumental task in astrophysics. Both fields require the use of advanced technologies, rigorous methodologies, and creative problem-solving. If you found this discussion helpful, let us know in the comments, and don't forget to hit the subscribe button. We have many other videos that cover a wide variety of topics on science and artificial intelligence.